Hello viewers, welcome to the CEC Gurukul live lectures. If you remember, this is the fourth lecture in our series on photosynthesis. We have dealt with at great length regarding the history of photosynthesis, the concept building of the equation of photosynthesis, the nature of the photosynthetic pigments, the nature of light itself, the discovery of the pigment systems, and the way light activates the reaction centers and the arrangement of the two photosystems on the membrane of the grana, that is either on the oppressed ends or on the non-oppressed ends. Today's lecture is going to be devoted to the cyclic and non-cyclic photophosphorylation, which is a very rich content topic as far as the newer ideas on the generation of ATP vis-a-vis the generation of ATP in oxidative phosphorylation is concerned. In addition, there are a whole new fund of information regarding the topography of the different types of electron carriers in the electron transport chain, which are responsible for the process of photophosphorylation. Basically, photophosphorylation is going to be the key process of light reaction, which along with the formation of NADPH is going to constitute the raw material for the process of the Benson-Calvin cycle or the dark reaction. Let us first begin with what are the key players in the light reaction. We know that it is the photosystems 1 and 2. We have discussed at length regarding these photosystems, whether they should be called as groups of quantasomes. Then we concluded no, and basically they are clusters of chlorophyll pigments, where the different types of chlorophyll absorption pigments of different wavelengths are arranged in a certain specialized manner, and some of them would act as antennae, the other ones as the collection centers, and a few of them as the reaction centers. In addition to the photosystems 1 and 2, there are a whole series of electron carriers, and these electron carriers are the ones which are arranged according to a series of the redox potentials, and they are the ones which are responsible for generation of ADP and NADP. These two, ATP and NADP themselves, are going to carry the electrons to the Benson-Calvin cycle. Water would be the main source because it is going to be the ultimate donor of electrons to the whole system. We would see shortly that water is going to be broken down or photolysed at the PS2. And then of course, the modern concept of ATP synthase, because merely saying that ADP plus inorganic phosphate gives rise to ATP is a very sketchy description of such a complicated process. This specialized enzyme has various structural components. We will see that it is a multi-enzyme complex, wherein there are different types of proteins and it behaves like a rotor or a motor. We have basically two types of photophosphorylation, and they are what we call as the cyclic and the non-cyclic type of photophosphorylations. The key players would be starting with the photosystem 2. As we have said in the previous lectures also, photosystem 2 operates at wavelengths less than 680 nanometers. So, that means basically it is a high energy system, it is a very strong oxidant, and a weak reductant. It is at this photosystem where we have the reaction center in terms of P680, the light quanta are absorbed, and finally water molecules get split into 4 protons, 4 electrons and oxygen. I need not remind my viewers that this reaction is perhaps the most vital reaction because of which we are alive today, because the entire oxygen is going to be released as a result of oxynogenic or oxygenic photosynthesis on the basis of splitting of the water molecules. Now, the electrons which are released are immediately donated to the chlorophyll molecules, because they would be losing their electrons in terms of photoexcitation. Oxygen is released into the atmosphere, and of course, the H ions are released into the lumen side of the thylakoid membrane. We would see subsequently that this creates a very strong electrochemical or a proton gradient, which is vital for the generation of ATP through the functioning of the multi-enzyme complex of the ATP synthase. The other component would be the electron transport chain itself, because once light energy has excited the electrons, the energy level of course increases, and so much of high energy is there that it has to be transferred. Now, I am reminded of an analogy. Suppose you have pizza or a cake being baked in the oven. How are you going to take it out? It is going to be extremely hot. So, you would definitely require mittens for doing so. Likewise, when we have the chlorophyll and the electrons in an excited state, they have to be carried by somebody, and that somebody is the electron transport chain, where there are various electron acceptors, and they are going to have a series of these electron acceptors which will pass these electrons to the different system, and that is to the photosystem 1. So, the key donor is the photosystem 2 of electrons, and finally, it goes to the PS1. And then another very important carrier would be NADP, which is the coenzyme 2. You know the full form, it is nicotinamide adenine dinucleotide phosphate. In case of respiration, it is coenzyme 1, which is NAD, and therefore, NADP is also a carrier for the high energy electrons. So, to say ATP and NADP are basically the mittens for high energy electrons to be transferred. In one of our earlier lectures, I had tried to make you appreciate that all these complexes which are present for the electron transport chain and the photophosphorylation for light reaction have a certain orientation. We have divided these complexes into 4, namely the complex 1, its proteins which belong to the light harvesting center 2, that is of the photosystem 2, they are directed towards the lumen, along with those of the complex 2, which is the cytochromes. On the other hand, the complex 3, which constitutes the light harvesting center or the photosystem 1, along with the energy coupling site, which is the complex 4, they are present towards the stroma. So, that means there is a specialized vectorial arrangement of these 4 complexes on the biological membrane. As you can see on your screens, the vectorial arrangement is very specific, because ultimately the NADPH is going to be formed at the stromal side, because it has to go and work in the Benson-Calvin cycle, which is occurring in the stroma. On the other hand, the electrochemical gradient is going to be created from inside to outside. From inside we mean from the lumen of the chloroplast towards its stroma. So, this is how the vectorial arrangement of the thylakoid membrane components or the complexes occurs. Let us begin with the process of light reaction as to how do the electrons start functioning. We have already learned that the photoexcitation has taken place, the funneling of the electrons through resonance transfer has taken place, and we find that the reaction center has already been activated. So, the first step of the photoexcitation would take place at the PS2. As I said before, PS2 is a high energy system, and this high energy system is going to operate at less than 680 nanometers. It is a strong oxidant. So, the first step would be when the light quanta or the photons, they strike the chlorophyll molecules, it is at PS2 that water is going to be split, and it is going to give rise to protons and electrons, and in the final process oxygen molecules are going to be released, which are going to be released to the atmosphere. This is the very important aspect of the donation of electrons from water. Once the chlorophyll has its photo excited electrons, they are going to leave the chlorophyll molecule, leaving a miss or leaving a gap in the chlorophyll molecule. Who is going to compensate? It is the water molecules which are now going to compensate by its splitting into H and OH ions, and this is precisely what Hill called as the photolysis of water. So, this will happen only in the presence of light. You can imagine it is electrolysis in the presence of light. There is no electric current, but it is the solar energy or light energy. Oxygen evolving complex is a very complicated one, which is located on the lumen side of the thylakoid membrane, and it is associated with manganese and chloride ions. So, ultimately 2 molecules of water, they break down into 4 protons, an oxygen and 4 electrons, and it is these 4 electrons which are going to have a different fate than the 4 H plus, because the 4 H plus have to make a concentration gradient of protons within the lumen also. So, it would mean the moment we have photoexcitation, the moment there is light energy falling on the chloroplast, the pH of the grana lumen is going to be highly acidic, because it is going to be full of H ions. So, that means the pH lowers and the medium becomes much more acidic. This water evolving complex is present at the PS2. The second step after the photolysis of water would be that the electron which has left the chlorophyll molecule and which has now been adequately compensated by water through its photolysis has to be captured by some other carrier. This is the mitten that I was talking to you about, and there has to be a primary acceptor which will now be present at a much excited level. That means at a much more minus redox potential, and this has been found to be a plastoquinone. Interestingly, plastoquinone is not a component which is bound to the plasma membrane. It is in fact a mobile component, and it can therefore move to either of the faces, that is the face towards the stroma or towards the lumen. So, it is a mobile element which is present. So, it is the primary acceptor, and once it accepts the electrons, it enters the third step. It cannot hold on to this electron for a long time, it has to now go through a downhill transfer, and this transfer of electrons now goes to the cytochrome complex. We should remember the cytochromes are iron containing electron carriers, which are going to function on the basis of ferrous ferric oxidation reduction reactions. And while going from one cytochrome to the other, we find that so much of energy is lost, which is sufficient to pump the protons against the concentration gradient from the stroma across the thylakoid to the lumen. So, that means more and more of the H ions enter the lumen, therefore, the pH is lowered further. The pH is lowered further. This is the basic idea of the second step. After the third step, now we enter the fourth step. You should remember there are enough H ions in the lumen, and the lowermost position of the electrons after the transference from the cytochromes is the plastocyanin. We should remember plastocyanin is a copper containing carrier, and the plastocyanin is also a movable element. So, these are the two mobile elements within the plasma membrane. All others are fixed, and they have a particular vectorial arrangement. So, plastocyanin is going to be in fact a connecting link between photosystem 2 and the photosystem 1. So, first or the initial 4 steps, they belong to the PS2, and the fifth step is the beginning of the photoexcitation of the photosystem 1. We should remember photosystem 1 operates at wavelengths greater than 680 nanometers, and its P700 is the sink, or what we call as the energy trap. And this photosystem also gets excited. Once it gets excited, it loses its electrons, and once it loses its electrons, then they are immediately compensated by the electrons which have come by the splitting of water ultimately and eventually, and they have now come to the PS1 through plastocyanin. That is why I said a moment ago that plastocyanin seems to be the connecting link between the PS2 and the beginning of PS1. The electrons, once they are again ejected, they are transferred to another transport chain, the second of the series, which is ferredoxin. Ferredoxin would be an iron containing mobile element, but it is present on the stromal side, as you can see in the diagram, and not on the lumen side. Therefore, this particular there is a reason behind this vectorial arrangement, because ultimately NADPH is to be formed, and as I said before, the PS1 is a strong reductant and a weak oxidant. This is in contrast to what we had described for the PS2. And the seventh or the penultimate step would be that the electrons are finally transferred by the enzyme NADP reductase, and NADP reductase picks up NADP plus the H plus and gives rise to H plus is already there through a series of reactions taking place in the first 6 steps, and ultimately gives rise to NADPH. The formation of NADPH is called as the reducing power. We will have a lot of references to the reducing power subsequently. And finally, the protons are pumped into the lumen. Why? Because by this time there is enough concentration gradient which has been created the concentration of protons is many times higher in the lumen as compared to the stroma, and they pass through specialized proton channels of the ATPase complex or the ATP synthase complex. We will discuss at length about this subsequently, and ATP is produced in the stroma. I would repeat, ATP is produced in the stroma, although the entire process is taking place in the grana. We always say that light reaction takes place in the grana and dark reaction in the stroma, but then on which face of the grana? That is, ATP is formed on the stromal side, because it does not have to pass the membrane to go and connect itself to the Benson-Calvin cycle. It is needed there, and so is the fate of NADPH. Having said that these are the 8 steps, we have a summary of the light dependent formation of ATP by a process which is the chemiosmosis. So, if we are asked to conclude the 8 processes which we have just seen, photophosphorylation again would be defined as the light dependent formation of ATP by chemiosmosis. Let us have the genesis of the word chemiosmosis, a chemical reaction which is going to be powered by osmotic principles or osmotic forces, which means it is the concentration gradient of protons which are going to generate. Now, where does then light come into the picture? We would see subsequently. To graphically make you realize as to the overall picture of photophosphorylation, we have the vectorial arrangement of the different components of the 2 photosystems, the electron carriers, the place for proton generation, and of course, the site for ATP synthesis. We find that we begin from the begin with PS1, and it is this particular phase with PS2, and this is the phase where the protons are going to be generated. This gives us a very important food for thought, and that is, suppose the PS2 is not there. If it was not there, then the answer is very simple. There would not have been any protons to react with in the final analysis with NADP, and if this was to happen, there would not be any NADPH. We would subsequently see what are the implications if this was to be true. Well, it is true in certain cases, which we call as the cyclic photophosphorylation. Ultimately, the ATP is going to be generated on the stromal side, but then the protons are going to enter the specialized proton channels of the CF0 and CF1 complex of ATP synthase. This is a multi-enzyme complex. Just to give you a final overview of the process of photophosphorylation, just to highlight the sites across the membrane where all the functions are taking place. Protons inside the lumen, which we call as the intermembrane space, the thylakoid membrane. On the outer side, we have 2 sites where the PS2 and the PS1 are getting energized by sunlight, and the ATP is generated on the stromal side. The different components which have been made into a graphic show us that the different types of protons at different sites, the electrons and the mobile elements, they are present on the 2 faces of the grana membrane. Let us come to a more serious question now, as to there should not be any need to contradict ourselves when we make a statement that photophosphorylation is the generation of ATP in the presence of light, which gives us a very logical question. is light directly required for ATP synthesis, or is it that light is triggering something else and then finally ATP is the product? What is the mechanism or the exact mechanism of the ATP generation, that is photophosphorylation? The word photo is there as a prefix. It is understood that light must be doing the vital role, but then how direct or how indirect? Then the other question which we pose to ourselves is, how is the process of photophosphorylation different from oxidative phosphorylation, which is occurring in the mitochondria? Because in both the cases ATP is the ultimate product. And finally, what is the recent thinking on ATP synthase enzyme? How does it work and how many ATP does it produce per electron or the proton gradients? The story for the mechanism was unfolded by a scientist, Peter Michel, who in 1961 gave the famous chemi-osmotic coupling hypothesis. I believe, like all other great scientists, his work was also ahead of time and perhaps was not appreciated at that particular moment. What he said was that the electron transport chain creates a type of a concentration gradient by pumping the protons into the thylakoid space. This is exactly what we have been discussing for some time now. And as these protons they diffuse back into the stroma, they are going to create some sort of a gradient, and this gradient is going to drive ATP synthesis, and it is therefore the proton gradient which is the driving force for ATP synthesis. Now, this idea was very much ahead of time. There was no very direct proof to this, and that this is the direction of the movement of the protons, and therefore this idea was taken with a pinch of salt. It was not before the German scientists Andre Jaganov and G. Heinz, they experimentally proved 2 years later that light is not of direct requirement for ATP synthesis. Of course, light works indirectly or in the initial stages, that is, it induces a proton gradient, and it is ultimately the proton gradient which is going to drive conditions which are responsible for the generation of ATP. And they performed a very simple and yet a very elegant experiment. They incubated chloroplasts in the dark, that is, they added a suspension of chloroplasts to a buffer which was at pH 4. Now, pH 4 means it was quite acidic. This buffer slowly penetrated into the internal compartment of the thylakoids, and therefore a stage came where the interior of the thylakoids had a very low internal pH, in other words a highly acidic medium. Then they added the entire experiment was done in the dark. Then they added ADP and PI, that is inorganic phosphate, to the dark suspension, and suddenly they changed the outside medium, which was originally 4 to 8, that is from a highly acidic to a highly alkaline, thereby creating a very large pH gradient. And this gradient was created, of course, momentarily across the membrane, and to their surprise they saw that the protons they moved out of the thylakoids into the medium along the concentration gradient, and this was the beginning of generation of ATP. Now, what a nice experimental proof to in support of Michel's theory, and it was this support which finally led people or forced people to take Peter Michel's ideas seriously. Eventually, Peter Michel also got a Nobel Prize, but many years later for this idea. So, that means the production of ATP in photosynthesis has to be through the generation of a proton gradient, and the gradient has to be from the lumen towards outside. And this led us to find out more as to what is the exact structure of the ATP synthase, because we know for sure that there are knob like particles which are present on the mitochondrial cristae, and they have been called as the Fernandez-Moran particles, named after the discoverer, the F1, F2 and the F0 particles. And we also know at great length about the electron transport chain which is involved in oxidative phosphorylation. But then do we have a parallel in case of chloroplasts? Have these two organelles evolved in a parallel manner? Well, that is a question which is within the purview of evolutionary molecular biology, but then it would be sufficient to find a contrast here between oxidative and photophosphorylation. We should remember that within the mitochondria a similar type of process ensues, from where we have the formation of ATP through the proton channels of the F0, F1 particles. The channels are there in the F0, and they are present in the matrix. The ATP is formed, and in the chloroplast the ATP is generated on the stroma side. The difference therefore would be that it is the sunlight which is running the show of the formation or the generation of ATP through the driving of the pump, and it is the oxidation of reserve food material or sugars ultimately which is the driving force for ATP synthesis in respiration. So, there is a unique parallel between the two processes, but then in terms of evolution these two things would have come simultaneously. That is a topic of further discussion. The current ideas regarding ATP synthase enzyme have revealed that they should be called, in case of chloroplasts, not as F1 or F0, they should be called as CF0 and CF1, because we are referring to the chloroplasts. It is also a multi-enzyme complex, and there are various coupling factors. The coupling factor CF1 is the hydrophilic component, and it is composed of various other components, namely as you can see on your screens, starting from alpha to epsilon. On the other hand, the CF2 or the coupling factor naught is the hydrophobic component, and also composed of A, B, B dash and C12. These are the different proteins. So, it is basically a multi subunit protein complex, and the CF0 has proton channels, because whenever there is a gradient, there is no other place in the membrane of the chloroplast grana or of the mitochondrial crista from where the protons can pass through. It is only through the proton channels that they will pass through, and ultimately there is no place they can go except for the catalytic sites of the CF1, where energy is released for the synthesis of ATP. So, we believe that this type of arrangement of the channels of the protons is going to be possible by the rotation. So, it is like a rotor which moves, and the CF0 has a rotor, and you can see there is a rudder on the upper side also, and it moves, and every rotation of this enzyme is going to generate 3 ATP molecules. So, this is one of the most fascinating aspects of bioenergetics and biophysics, of course, where this type of a mechanical rotor is seen to be responsible for the generation of energy. We come to the Z scheme of electron transfer, because having a knowledge of the photophosphorylation, we should now try to understand whether both these photosystems work at tandem, or can there be situations where they work alone, and in the latter case which one of them can function independently. Why do we call it as a Z scheme? Because the arrangement appears to be like a Z. Americans call it as Z. The Z scheme was initially given by Hill and Bendel, and in the recent times most of the work has been done by Professor Govindji, who is an Indian American scientist, recently retired from the University of Illinois, Urbana-Champaign, and he is the world authority on the Z scheme, discovered so many of the factors, so many of the intermediate cytochromes and other aspects, the other nuances of Z scheme. The scheme can be either cyclic or non-cyclic or pseudo-cyclic. Cyclic would mean that the electrons which have been excited have to come back. Non-cyclic would mean that they would run away. By running away, we mean that they would combine with some other component and they would never come back. If they would never come back, then they would require continuous replenishment, either by the splitting of water. And there is a pseudo-cyclic photophosphorylation, which is a very unique type of a primitive type of photophosphorylation occurring in certain bacteria, where there is no involvement of NADP, and it is basically a FMN run show. Let us try to appreciate a Z scheme of photosynthesis, where we find that the PS2 gets excited first, the electrons go to plastocyanin, and then by a series of the descending series of the cytochromes, the cytochrome B6 complex, 559 complex, they come to PS1. PS1 is photoexcited itself, and its electrons, once they are ejected, are promptly accepted by ferredoxin, which goes through again a series of the second electron transfer to the NADP reductase complex. By this time, the electrons and of course the H plus, which were there by the splitting of water, they combine to give rise to NADPH. The basic idea therefore would be that the two photosystems are running here at tandem, that is together simultaneously, and the two products are ATP and NADPH. In one of our first lectures, we mentioned about the terminology. The formation of ATP here is called as photophosphorylation, and the formation of NADPH is the reducing power. Summing up both of them, it would be called assimilatory power, and a complete assimilatory power is required to be transferred from the grana to the stroma for running the Benson-Calvin cycle. If we were to just graphically depict the non-cyclic photophosphorylation, because we have just seen that the electron which comes goes and ultimately gets consumed by NADP to give rise to NADPH. So, the two forces which are running here are at PS2, which is again through the photons, and the other one at PS1. So, that means the two steps, both of them are running together, PS1 as well as PS2, and this would result in the formation of a complete set of assimilatory power which is required. This is normal photosynthesis. What would happen if one of the photosystems doesn't work? We asked ourselves a question some time back, which one? So, it is always the PS2 which can never run independently. In the absence of PS2, PS1 can run independently. The question is the naming of these systems itself. PS1 was named because it could run independently and was perhaps the first type of a set of chemical reactions which evolved in organisms. As we can see, if PS2 is not there, that means the high energy radiations would not be there, and only wavelengths greater than 680 nanometers would be available. So, PS2 would never function, and if PS2 does not function, then PS1 gets excited and it goes to the NADPH. As I said, the electrons will run away. Now, there is nothing or nobody to elope with. With whom would they run away? So, they will do nothing, but they would have to come back. So, that means this is the basis of cyclic photophosphorylation. I have tried to oversimplify the process. Actually, the process of cyclic photophosphorylation is also important in metabolism, because there are situations where NADPH is really not required, and if it is so, then if a cyclic photophosphorylation can give us only one ATP, it would be good enough. So, that means Calvin cycle itself is going to use more of ATP than NADPH, because if we see the equation, it is 18 ATP versus 12 NADPH. So, there are situations where if wavelengths are not available, the desired ones, then only cyclic photophosphorylation ensues. So, to summarize, cyclic electron flow would take place only when the PS1 is operative and PS2 is not operative. The protons nevertheless will be pumped, ATP will be formed, but then since there are no protons available for the NADP and of course, the electrons, because electrons have to come from there, and there will be no NADPH. I must remind my viewers, the equation can be said, or the summary, as no PS2, no water splitting, no protons, no electrons, and therefore, no sequence of replenishment to PS1. This is the basic idea. Purpose of cyclic electron flow could be that if by chance there is an increase in NADPH levels, so there could be a sort of a feedback where not much of non-cyclic photophosphorylation is required, and the machinery of the chloroplast could now go to the mode of a cyclic electron flow. But mind you, this is only temporary. So, the cyclic electron flow is not going to involve PS2, nor would it involve the formation of NADPH. Let us try to connect what we have just said with our second lecture, when we were talking about the concepts of red drop and immersion effect. Basically, immersion effect is nothing but non-cyclic photophosphorylation, where a perfect Z is formed, both PS1 and PS2 are operative, and ATP and NADPH, which means a complete assimilatory pair is formed. And in the event of the absence of PS2, because there was no shorter wavelength, no photolysis of water takes place, no H plus, and only PS1 is operative. As a result, ATP is formed, but then NADPH is not, and this results in an immediate drop in the quantum yield, and this is called as red drop. Pseudo-cyclic phosphorylation is the third possibility. This was discovered by D.I. Arnon in 1954, and as I said before, it does not require NADP. So, it is actually an oxygen dependent FMN catalyzed photophosphorylation. Here it is, the whole show is run by FMN, and FMN itself is an auto oxidizable hydrogen acceptor. It will break the water molecule in the presence of chlorophyll, and it would produce FMH2 and oxygen. Now, the question is, how would the ATP be generated in this case? ATP would be generated from ADP itself simultaneously, and during the process the auto oxidizable FMN can be reduced by itself, and it will be oxidized back to oxygen to release FMN and water. So, pseudocyclic photophosphorylation occurs in certain specialized bacteria. Another very important aspect of the knowledge of the cyclic and non-cyclic photophosphorylation would be, if we want to kill a plant, we have to use a herbicide. Now, where are the herbicides going to act? Are they going to at the physiological level? So, two types of herbicides are available. One is DCMU, which is also commercially called as Dayron. This is an inhibitor of non-cyclic photophosphorylation. You remember, the cytochrome B complex, 559 complex, is the connecting one after the excitation of PS2, and it is to send the electrons to PS1. If this particular site is blocked, no electrons will pass to the PS1 and photosynthesis will not take place, which means the complete assimilatory power will not be made, and as a result the plant would be ultimately starved and it would die. Another category is of paraquat, which is also called as the methyl viologen. It will block the electron flow not between the PS2 and PS1, but it at the stage where the second electron transport chain is operative before the NADP reductase complex. It will itself accept the electrons rather than the NADP doing so, and as a result it will prevent the NADP from getting the electrons. So, that means it blocks the electron transport near the PS1 ending. So, this is in both the cases, if any of these sequence is blocked, the photosynthesis goes. So, that means in order to summarize, the basic idea would be that for every oxygen molecule evolved in the light reaction, 3 molecules of ATP and 2 molecules of NADPH along with protons are produced. So, that means if one looks at the equation, the final equation, it is 24 molecules of water plus 18 molecules of ADP would ultimately generate 12 molecules of water and 18 ATP. You remember, all these ingredients, that is 18 ATP and 12 NADPH, are actually required for the Benson-Calvin cycle, which is the topic for our next discourse as to how these NADPH and ATP in the form of assimilatory power, they go to the stroma for running the Benson-Calvin cycle, because without this the dark pickup of carbon dioxide is not possible. So, the basic feeding part is the light reaction, which now connects to the Benson-Calvin cycle or the C3 cycle.